leukotrienes are an inflammatory mediator, and there's several classes of them. And what we found with the fish oils is that they changed the leukotriene profile to the less inflammatory type of leukotriene. We call them leukotriene B5s versus the leukotriene B4s. And that has the potential for decreasing inflammation.
Well, welcome to the Pet Food Science Podcast. I have the privilege and sort of fun opportunity to have this Dr. Gene Hall, Oregon State University. Dr. Hall is an expert on fatty acid metabolism, and we've worked a lot on omega-3 fatty acids, and so we're going to talk about them today. So with that introduction, Dr. Hall, I wonder if you could tell us a little bit about yourself and how perhaps you got interested in omega-3 fatty acids.
Thank you for your kind introduction. I'm happy to be here visiting with you today. Well, as you know, I got interested in working with N3 fatty acids, also called omega-3 fatty acids, about, it was in the early 1990s, so maybe 4 decades ago now.
I don't want to talk about it.
And I was working with you. So I don't exactly remember the exact moment of how we connected, but there was a common interest, and I had facilities, and we wanted to look at dogs and pet foods and how we could improve pet food and nutrition and the health of pets. And the idea of N3 fatty acids being added as a supplement to the food sounded very intriguing to me. What I knew from human literature, and even my postdoc in nutrition at the Oregon Health Sciences University, where they were studying N-3 fatty acids, or the omega-3 fatty acids, had prepared me for that. And it just seemed like a golden opportunity to work on this same topic in dogs and eventually in cats. And that's where we all started, early 1990s.
Corbion's Algaprine, DHA-P3, the algae-based omega-3 DHA to boost brain, cognition, immune system, and well-being. Adding it to pet food is easier than ever. Cut out the middle fish and go straight to the natural source to enjoy all the benefits of omega-3 DHA. It's better for the planet with zero impact on the ocean and low carbon footprint, and it's easy to use. The highly concentrated biomass powder is suitable for multiple applications. Learn more at corbion.com.
Yeah, I wonder if we might just sort of do the chronological walkthrough for a minute. So one of the things that I remember we first started with was sort of looking at, if you feed them, do you find them in the blood, and then looking at ratios. Remember how the ratios were really kicking around? Let's talk as an introductory way, as a base. Let's talk that first series of studies that you did, or we did together.
That's really interesting, because I don't know exactly how we knew how much to add initially. And we thought the ratio, in fact, if you looked at the ratio in the pet foods, that's probably where we started. It was quite high in the N-6 fatty acids. And maybe I should digress just a bit to explain. It has to do with where a double bond is placed in a polyunsaturated fatty acid. It seems like a very simple chemistry change, but the location of that double bond, be it in the 6 or the 3 position, made a whole difference in the classification of the fatty acids and then the effect in the animal. So we started out with what was normal, a lot of corn oil or more plant-based oils in the foods, and just started changing that ratio a bit and adding more of the fish oils or the fatty acids from fish oils into the food. And the numbers probably don't mean a lot, but I think we had about a 30-to-1 ratio of N6 to N3 fatty acids initially, and we went all the way down to like a 1.5-to-1 ratio, which meant we added a whole lot more of the N3 fatty acids. And that's kind of the early studies. We then started looking at differences, comparing groups of dogs fed one diet to groups of dogs fed the other diet, and how that affected the change in the serum fatty acid profile. That was the early work, trying to decide if it was the actually the dose or the ratio that was important?
And remember, we did what I always thought was sort of an elegant study where we kept the ratio the same and changed the dose, because previously we'd been looking at really changing both. Dose would change, and then that would force the ratio to change. And then we did that one study with dogs where we changed dose independent of ratio.
Absolutely. So just if I can explain what that means, we kept the ratio the same on all of our diets. I think we had 3 or 4 diets, and you can make the same ratio with 3-3 or 6-6 or 9-9 or 12-12. So the ratios all looked the same, but the dose was quite different. And what we found was that really up to a certain point, it was that the dose was the most important. So changing the amount of N3 fatty acids that the dogs were fed, regardless of what that ratio was, up to a certain level was really important. And so the fact that we were just providing a new source of fatty acids from fish oils primarily was really important.
And it's interesting we use fish oils because that's what's available or was available at the time in the marketplace. And intuitively you'd think, well, the fish must be making it, but the fish don't make it. The algae that they eat, and then of course being the good monogastrics that they are, they're storing the fatty acids that they eat, and they end up being high in these omega-3. So one could imagine, moving that food chain a bit and getting algal oils. But at the time, we never had them available. We never used them.
Right. So our early studies then, back to your theme, was can we change the amount of N3 or omega-3 fatty acids that we feed dogs? And we found that was yes. And then we started looking at what the effect on the animal was. And we had ideas from the human literature that it was affecting the immune response. Mm-hmm. And sure enough, we found that the additional N3 fatty acids actually depress the immune response or depress the cell-mediated immune response. And you always want things to go in the positive direction, but with immunity,
unless you happen to have psoriasis, of course.
Yeah, with immunity, sometimes less is better. So that was really interesting to me that you could impact the immune system just by changing the type of fatty acid in your diet.
Well, it's funny because the pets and the mammals have this ability to elongate and desaturate at some level, and they certainly have the ability to selectively metabolize, and yet what you feed them really matters. And that's why these omega-3 fatty acids get to have that effect.
Right. So a lot of our studies then, after we figured out that the dose was very important, again, at levels we used, it might be the ratio became more important. If you feed more and more, then they're going to compete with each other. And what you're actually doing with these N3 fatty acids is incorporating them into the cell membranes.
Yeah, let's talk about that.
Cells. And so it could be all body cells because they all have fatty acids as part of their bilipid cell membrane. And so we would then take cells specifically from the blood and study them to see what their immune functions, how they were affected. And one of the cells we used were the neutrophils because we knew they are important in bacterial killing. So if you get an infection with a bacteria, the neutrophils are important in that. So we started looking at how the fatty acids that we fed in the diets affected neutrophil bacterial killing and some of the genes that were associated with the enzymes that they used to cause the bacterial killing. And sure enough, the fatty acids could affect neutrophil bacterial killing. And that was really important because we also, at the same time, were looking at how these immune function tests changed with age. Remember, we looked at puppies and adult dogs that were in the what we would call the middle range, maybe 1 to 5 years of age. And then we had the group that we called the senior adults.
Right.
I don't think we got to the geriatric side, but they were over 8 years of age. And what we found was that with age, they had decreased neutrophil phagocytosis or bacterial killing. So the older dogs weren't able to get rid of a bacterial load. This was in cell culture, but we assumed that the same might be true in the animal.
It's a good marker. I think so too.
Yeah, let's try this in dogs after we've fed them the N3 fatty acids. And we were able to offset some of those age-related changes. And that was exciting to me.
Very exciting. In fact, it sort of suggests you get a mutual benefit, right? So we saw this good benefit in being able to kill those nasties when they're coming across the barriers. And then at the same time, remember that first work we did, I believe, Dr. Wander, or we would say Rosemary, was the first author. We saw the leukotrienes go down, and then later we saw the thromboxanes go down.
Right.
So we had a couple of good indices of inflammation going down, but yet still enhanced or at least maintained this ability to kill the bacteria. Can we talk about that a little bit?
So some of the mediators of inflammation are leukotrienes. People may have heard of those, particularly with all the allergies around there. There's a lot of newer medications that are blocking the effects of leukotrienes, but they're leukotrienes are an inflammatory mediator, and there's several classes of them. And what we found with the fish oils is that they changed the leukotriene profile to the less inflammatory type of leukotriene. We call them leukotriene B5s versus the leukotriene B4s. And that has the potential for decreasing inflammation. So in that early study that you mentioned with Rosemary Wander as a co-author, we showed that leukotriene B5 levels go up, or the ratio of leukotriene B5 to B4 is increased. And that would be a potential benefit for decreasing the inflammatory potential. And interestingly enough, we showed that in dogs in that study, but I also showed that in horses. We did a horse study where we fed them fish oil or corn oil, and the same was true. We took the neutrophils out and looked at the leukotriene profile, and it also showed an increase in the leukotriene B5s, the less inflammatory type.
So it really suggests you have a mammalian response, right? Because those herbivores, they're not set up to have many omega-3s in their diet, well, at least not from fish oil.
Not even just mammals. We did it in poultry chicks, too.
Oh, interesting.
I don't know if you're aware of that study, but we did.
I'd forgotten that.
Yeah, we did thrombocytes from growing chicks, and their mothers had been fed a heavily fish oil enriched diet versus a corn oil enriched diet. And those same thrombocytes produced less of the leukotriene B4 and more of the leukotriene B5. So, yeah, it seems to be a phenomenon in mammals and birds. So.
Pretty conserved, yeah. It's pretty fascinating. Mm-hmm. And remember, we sort of went on to the thromboxanes in that study we had where we added the omega-3s and the urinary thromboxane went down. That's sort of an interesting marker as well.
Absolutely. So we looked at lysophospholipid, which is a class of lipids, and showed that there were changes in the urine, you're right, associated with feeding of the N3 fatty acids. So it seems to be an effect that happens to all cells in the body because we're changing the fatty acids in the membranes. And then those lipids in membranes are what get clipped off and reacted on by enzymes that produce inflammatory mediators. So it makes sense if you change the starting material, you're going to have a different inflammatory mediator. And hopefully with the N3 fatty acids, we have a less inflammatory mediator.
Well, we never measured it, but we've heard the anecdotal things that when you add these omega-3 fatty acids, that the skin looks better, the coat shinier. What do you want to talk about that? Is that related to inflammation or?
Absolutely. It's very hard. We call those the soft metrics.
No pun intended with the soft, shiny skin.
Exactly. It's not as easy. Although I think we did have one study. We've done our studies, as you know, in both client-owned pets and in colony pets. And the client-owned pets did kind of talk about their skin quality.
Yeah, I think we did. I'd forgotten that. In the client-owned pets where we added the omega-3 fatty acids, I'd forgotten that one.
Yeah, and again, it's a little more difficult because you have lots of different owners describing what they think of their pet's skin. their hair coat color, luster, softness, dander production. But in general, we did think that the owners did think that they had a better skin. And I don't know about you, Dennis, but I take fish oil and I feel like my skin is softer. Again, it's one of those metrics that's an opinion based metric, but I do think it's real.
Yeah, how value is an N of one when the subject knows the treatment.
Exactly.
But I'm still using it, so it must be good.
Exactly. And like we said, the owners in that particular study thought there was less dander, their hair coat had more luster, their skin felt and looked better. So, and we do know that N3 fatty acids are used in dermatology.
And I do want to sort of point out that study was a good randomized complete trial where the owners did not know the treatment, so
Right.
they didn't have the chance of going, yeah, it's fish oil, I bet it's good. They just got a colored bag, and we were objectively able to measure that response, although it's a subjective response.
Yeah, absolutely. So yeah, you're right. We've looked at what would I say, more measurable metrics to assess the immune system. The leukotrienes, the prostaglandins, the DTH, or the delayed type hypersensitivity reaction, the lymphocyte count. Remember, we saw in one study that the lymphocyte count, we can look at CD4 positive and CD8
Yeah, I
positive
remember that.
T lymphocytes, and you can actually decrease the number of CD4 positive lymphocytes. And if you add in the antioxidants, the vitamins E and C, you can increase the CD8 population. So we can effectively change that ratio, which will have an overall effect then on the inflammatory response. So we've shown by a number of tests, and then just the DTH, I think I mentioned that reaction. It's more where you give them an injection of an inflammatory substance right under the skin, and you see their reaction to that. That's all suppressed.
The audience might not know, but that's like if you're having a allergy response, a skin test for an allergy response. It's that type of test, and of course, sort of a objective measure again of that immune response and how inflamed and how, if you're looking for perhaps a pet that has this excessive response to dander or whatever, that you might want that to go down.
Yeah, and I should point out, everybody always thinks that a robust immune system, you want to have the most active and the most sharp, ready to go react. Is not always a good thing. Many of our infectious problems are exacerbated or made worse by how bad or how much the system reacts to that pathogen. If you can kind of quiet down that peripheral response, then they can get things under control. So we're not wiping out their ability to kill bacteria or respond to an inflammation. We're just kind of quieting down that overall fire, if you will, so that they don't have as many side effects associated with the inflammation.
Really a beautiful biological system that we are enhancing in ways that are probably beneficial with omega-3s. But then it's always a patient individual on top of it. Hey, I wonder if we could talk a little bit. It's a little bit of space, but you remember Dr. Zicker and I, we ran a DHA study where we had the DHA in puppies and of course had that neurological response where they basically learned faster. Can you talk a little bit about omega-3s and neurology and sort of, we've been talking immune response, but maybe just a little of your basic knowledge of, why that might be important for brain development or function?
Yeah, that's really interesting that you bring that up, in that a couple of the body tissues that have the highest concentration of the N3 fatty acids in their cell membranes, interestingly enough, are the brain and also the testes. And I don't know exactly why those two organ systems or tissues are so heavily enriched in these N-3 fatty acids, but you have to kind of assume it's there for a good reason. And I remember early on, that kind of brings me back to like this 4 decades ago, they were looking at in monkeys during my postdoc at the effects of N3 fatty acids on early development in monkeys, thinking of it as a model for humans. And visual acuity was altered, or they could measure changes in that based on how much of the N3 fatty acids were in their diet. It was altered in a good way. They developed better visual acuity. So I think of that as a brain function. And that's kind of how I was initially interested in it. So it didn't surprise me. I wasn't involved in your studies where you fed it as a brain supplement. It's kind of to older dogs, if I remember, to help them in
We
their
did it
senior
both.
years.
Yeah, both senior and in the younger puppies. The little beagle puppies are so much fun to work with. They become beagles, but when they're puppies, they're like the cutest
Yeah.
thing alive.
Well, what I remember was that the kind of walking off and staring into space and not paying as much attention in the geriatric pets and not having as much human interaction, all those were affected in a positive way by including more of the N3 fatty acids in their diet. And it just made sense to me that we had seen this in the monkeys in the early development, that visual acuity was improved. To the extent that I believe the people that I was working with in my postdoc were able to convince the industry that made breast milk replacements or powdered milk, if you will, for feeding infants to make sure that they included the N3 fatty acids in those because they were so important. So they recognized that in human medicine as well.
Thank you. I think DHA is really a good idea for that milk replacement. I don't know if it's required. It's not my space, but it's certainly an optimum would include DHA for sure.
Yeah, so including it on the young end when they're developing brain function, and then including it on the geriatric end when animals, in our case dogs, become what we call more senile, but maybe that senility is reversible to some extent, or at least you can prolong the happy years or the good years before that sets in. It seemed to be important with including more N3 fatty acids in the food.
So we've sort of talked about inflammation. We've talked about the value for skin and coat. We haven't talked about arthritis, another space where N3 fatty acids seem to have, and you might expect that from what we've said, but really good data showing that when N3 fatty acids were added, they aid in the management of, joints. Could you talk about that?
Right. Now, I don't think I was ever involved in any of the joint studies, but I'm aware of that work. And I know certainly that you were involved in those studies. So arthritis, as we age, we all know that joints become more stiff and have more inflammatory reactions associated with them. And it was beneficial to feed them the N3 fatty acids. It's the same principle. You're decreasing inflammation and hopefully, that makes the pet feel better with the decrease in inflammation, and then their arthritic changes or their old aging joints are able to function better. So, yeah, I'm not surprised. We've seen benefits with, like you mentioned, the brain, with the skin, with the joints.
I wonder if we could look out a bit. We showed some benefits. Remember that one of our recent studies where we were looking at, now that was a spectrum of fatty acids, but it was on stone formation. Do you want to talk about that at all?
That's one that I missed. So in cats, we were looking at N3 fatty acids. Actually, we were looking at polyunsaturated fatty acids. So both the N-6, but the long chain, highly unsaturated, we call them polyunsaturated fatty acids. And we were looking at those in cats to see what effect they had on renal function. And we all know as age progresses, the glomerular filtration rate, which is the rate at which we form urine, slows down to the point where eventually kidney failure becomes a problem and animals can succumb to that. So our goal was to increase glomerular filtration rate and decrease those uremic toxins that are retained as a result of decreasing the filtration rate. So we were able to show in cats in early stage, or maybe they were just geriatric cats, that we could increase the GFR.
I think they were out of stage 1, yeah.
Yeah, they were the earliest of the stages of renal disease. So we could increase the GFR back to what looked like more adult cat levels, and we could decrease the SDMA, which is a marker associated with uremic toxins that are supposedly secreted through the urine, those levels went down. So those were both beneficial in older cats to preserving kidney function as they aged. And then as a sideline, we found out that they secreted less calcium into the urine. And we have a test that measures their potential for forming calcium oxalate stones. And that's a problem in cats, just like it's a problem in people, that they form stones and they can cause obstructions. And those aren't good for the cat. And in fact, we had that one study where we showed that cats with stones had a shortened lifespan by about 3
died
years.
Earlier from all causes, right? Not necessarily from kidney disease, but all causes. Yeah.
Yeah. So if we could decrease the stone forming phenomena in cats with changing the N3 fatty acids in food, that's also a big boost. Not only do we help out their kidney function, but we decrease stone formation. So I think that was a surprise to me that we could even go to that level.
I often think of the beauty of biological research is the window opens up and you can see inside, right? And you go, wow, I didn't know. I mean, I ran the study because I didn't know, and here it is. It's really, really kind of fun.
It is. And sometimes, as you know, we've been talking about fish oil, but we go on fishing expeditions and think, oh, well, let's just take a peek at the urine and see what's happening there. Not really understanding that we might find something, but just to maybe, wow, we didn't even think about looking there. And then we do find something. And that study that in cats where we looked at stone formation, if I remember correctly, Dennis, you can help me, but it wasn't just the N3 fatty acids. It was both fatty acids.
Yes, we should talk about that a little bit because we use available ingredients, right? So lots of times our studies, and in fact, we don't know which specific fatty acid, we think DHA in brain, okay, it's neural, but in fact, we're feeding fish oils or we're feeding a multiple mix of fatty acids.
Right.
And we sort of think, well, lots of times it's this specific one, but we don't know.
Yeah, actually, maybe I should back up and explain that. When sometimes I use fish oil interchangeably with DHA and EPA, but fish oil is just high in these two fatty acids that are commonly talked about as representatives of the N3 fatty acids. But icosapentaenoic acid, much easier said EPA, or docosahexaenoic acid, much easier said DHA, are just two of the major players in the N-3 fatty acids. And they're both in high concentration in fish oil. And you're right, we don't feed in our studies just EPA or just DHA. We feed commonly fish oil mixed into the food, which my understanding, at least with my cat, is they love fish. And so adding this fatty acid supplement to the food is not a detriment or a turnoff to the animal at all. They love that addition. And so we do get the benefit of having multiple N3 fatty acids when we use the fish oil. We get the benefit that they like it, but we don't know exactly which individual fatty acid is causing or responsible for the effects.
And, if we were to replay this in today's world where we have the algal DHA, I think we probably would have tried to narrow in, is this a specific fatty acid response? We just don't know. Right now, I think because, people are somewhat concerned about the sustainability of fish oils and they don't make the omega-3s, moving it back in that chain kind of makes sense. We just didn't have it available, so we didn't use it.
Right. Well, you might help me out on this, but sourcing fish oil, it seemed to me like fish oil was a byproduct of the fishing industry and that the sources went on forever. But is there a limitation in how much fish oil is it?
all things are limited, but people are worried about it. That's the great thing about pet nutrition. We often use byproducts so that, and there's nothing wrong with the byproduct. The primary ingredient screen is going one way, and then this stuff is left over, and we certainly have a benefit about feeding it to pets. That's better than landfill or something. But in general, I think overfishing is kind of a worldwide concern, and so at some point, maybe it matters. I'm not a supply guy, right? I'm a nutritionist, but we can see those worldwide trends and perhaps benefit to.
Well, certainly, if you could make EPA or DHA in the lab, I don't even know if that's possible.
But they do. They have algal production of them.
Oh,
absolutely.
Okay. I don't know how it would compare cost-wise, but that would be a sustainable then ability to produce the fatty acids.
Yeah, as nutritionists, we kind of don't play in that space, but I think it's an interesting one. And as pet food scientists, that's something to look at Horizon. And I wonder if we might talk about that in general. You're an expert in the field. If you were to look out and say maybe someone entering the field are interested in the metabolism of these omega-3 fatty acids, where do you see next steps might be, or what might you sort of, if someone to say, I was going to design a PhD program, Dr. Hall, what should I be looking at? Any suggestions?
Well, I feel like we've been on a fishing expedition, and we've looked at a lot of different things, and that's kind of true for what's been done in the field. I mean, we've looked at skin effects, we've looked at cellular effects in the membranes, we've looked at tissues like the brain and found out where the high concentrations are. We've looked in the urine, we've looked at renal function. So we've just looked at a whole bunch of things. But like you said, narrowing down the exact mechanism and which fatty acid is responsible for it, you could pick any one of those areas.
Sure.
Some of the areas would be more difficult to study than others, more invasive, I guess. If you wanted to really look at how the fatty acids incorporate into brain cells and then cause specific effects, those kind of projects get very invasive and maybe not as easy to do. If you wanted to look at kidney function, how exactly do they modify the different glomerular cells or tubular cells, and what are their overall functions that are being changed? Very mechanistic, again, more invasive, and maybe you have to take some of that work into tissue culture and see exactly where the effects are and how they're changing lipid membranes and what that does to different enzyme functions in those membranes. So yeah, you could become what I would say is more mode of action or mechanism of action at the cellular detailed level. That would give us all answers.
Yeah, good answers. Those are fascinating areas. I wonder too, I was, we've done a lot together, Dr. Hall, but the AGXT2 and started thinking, there's got to be a genetic component of how these things work together, and maybe that drives to some sort of this animal, this pet will get a benefit, this pet maybe not. So what do you think?
Right. So looking at gene expression related to the types of fatty acids, that's a really important. And we did just a little bit of that in these bacterial killing.
Yeah, we did. I recall that.
We looked at myeloperoxidase. I'm trying to remember our other genes. IL-8 receptor gene was one of the other ones, and an interleukin converting enzyme. I think those were the three genes that we looked at. And those were all, the gene expression at the messenger RNA level were all altered by the diet, the N-3 fatty acids that we fed. So we did get a little bit of insight into how our diet was causing a change in bacterial killing by our neutrophils. And those are all really interesting. It makes sense to me, but you're right that the N3 fatty acids are so integral to membranes and how membranes function, and the enzymes are in the membranes, that they could express there's enzymatic functions differently, or even at the nuclear level, they could change how DNA is expressed into messenger RNA. Those are all fascinating areas that we don't have a lot of information for.
There'll be a lot of opportunity for young scientists if they wanted to stay in this omega-3 fatty acid area and help us understand what happens, why, and then, of course, perhaps a chance to do it better, maybe by.
And so many of the things that we're looking at in the dog and the cat are also of concern for humans.
Sure.
The oxalate crystals in the urine of the cats that we showed were affected by the fatty acid concentrations in the diet. The inflammatory joint disease that we showed was helped by the N3 fatty acids in the dogs. The skin issues that we dealt with in pets, that people also have skin issues. So I see a lot of potential for funding is always the concern, but if you can relate it to a similar condition and our pets are just one of the benefits of helping to solve a human disease is that we're also benefiting our pets, even though my primary interest was the pet. So it's fun.
Well, Dr. Hall, thank you. I think this has been a very interesting discussion, and it's certainly a opportunity for pet food scientists to think about using these omega-3 fatty acids in food, and as we've discussed, lots of application for future research as well as our own foods that we think about what we
carry on our legacy as well. So we would enjoy seeing what they can do with that.
Thank you. Yes. Well,